Good day, my name is Heinrich Ferreira, and today I'm going to give you another walkthrough through the features of the Visual Genome Browser by using an example protein that is very well known, and that protein is RNA polymerase 2. It is a DNA directed RNA polymerase in the sense that it's directed towards DNA and it copies the DNA into RNA, and this process is known as transcription. These transcripts of RNA are then stitched together in a process known as splicing to generate a messenger RNA, which is exported from the nucleus and then used by the ribosome to create proteins by attaching the correct amino acids in the right order. So the gene I'm going to look at is Pol R2A, and the way you load it in the Visual Genome Browser is simply typing in the name in the gene search box. So we go to the Visual Genome Browser. It immediately highlights the box that, so you can just start typing Pol R2A, and then it gives you like a description here. You can magnify this by using this mouse wheel with control, and you can make it bigger. If you press enter on it or you click on it, it will navigate to the gene in the genome. You can see here it has just opened up chromosome 17. There's quite a lot to absorb on the screen. What you see here below the mouse is all the different alternatively spliced transcripts of that gene. In the info panel, you can see a description of how long it takes to approximately transcribe the gene. If you take a transcription rate of 35,000 to 10,000 bases per minute, you've got your main genome view, and if you move the zoom DNA view, it actually shows you here with a tooltip where exactly that gene can be found on the chromosome. And then you could also see it here on the right in the zoom gene view. You can zoom in with the mouse wheel. There is a way to actually filter genes in the Visual Genome Browser. Is when you click on this filter field, then it has this gene symbol filter. And if you click on that and you just start typing and you say polymerase R2A, it will filter all the genes that starts with this name. So if you press enter, it's going to filter this view so that you can only see that specific gene. This makes it much easier to distinguish where it is on the genome. You can also make this panel a little bit bigger. So what you see here is it's a color-coded view of the DNA. If you right-click on this panel here, you can see a color legend. So it shows you that adenine is blue, thymine is black. So those two bases are the ones that are bound to each other with two hydrogen bonds. Then guanine and cytosine, they are bright colors, yellow and orange. And then it also shows here that the start codon is indicated in green and the stop codon in red. And then the actual codons are mapped onto the zoom DNA view, and each of them have a different color. If you want to know what the specific colors means, you can go to the genetic code table. You can see here that hydrophobic or non-polar amino acids are this pink color. Then you've got polar uncharged, polar positively charged, polar negatively charged, and then stop codons. So if we go back and we zoom in a little bit, you can more clearly see everything. So there is actually a way to navigate from exon to exon. Each of these represents an exon of the gene. The first thing you must establish is on which strand of the DNA it is. If you look here on the info panel, you can see it's on the positive strand. And that means that the splice signals that is used by the spliceosome to stitch them together would be GT and AG. And each of the exons are stitched together using these splice signals that are part of the introns. So what is between the two, this is an exon, this is an exon, what's between the two represents an intron, and the introns are removed by using these GT-AG signals. So if you right click on this, then if you go to the specific gene, and then there's this option that says jump to the first exon and use this arrow greater than and smaller than keys to move between exons. So then it goes to the first exon, and you can use the keys to jump between them. So what we see here is the 5' untranslated region. Then you've got the ATG, which is the start codon, and then you've got all these different amino acids. If we navigate using that greater than and smaller than keys, you can see that this is the first exon, then the next, and the next, and the next, and each of them is separated by an intron, which is demarcated with GT and AG, GT and AG, and you can also zoom in to see it clearer. You can see that this is GT and AG, which is the splice signals that is used by the spliceosome to recognize exactly where the exon or the intron boundaries lie. Okay, so you can see on the left, you can see here on the left, this is the different transcripts. And if you want to switch between different transcripts, you can use the plus and minus keys. So if we press the plus and minus, you can see here, this is a different transcript, and then the next one. So this one is showing the colors. So this one, in this one, you can see the different codons clearly. If you go to the controls, you can click on this button over here, and then there's this option codons. Now if we zoom in a little bit there, you can see that there's different options for codons. If you click on aminos, now you can see that it actually shows you the first letter of the symbol for each of the amino acids, and that makes it much clearer which amino acids we are talking about. And then if we switch to amino color, what you can see now is it actually puts in the amino acid abbreviation in here, which is very handy to have. So now if we toggle this same checkbox from amino color to that view, there's different representations of the same amino acids. You could also switch off the codons view if you check the no codons button checkbox, and then you can simply just see the gene which is color coded to the same color as in the main genome view. If we go back to codons and we right click on this, it gives you this menu option where you have different options depending on what you want to do. You could copy the specific position, which is this position, to the clipboard. You could show more information in a window. You could locate the chromosome, it on a chromosome or the sequence. So let's just go back to the bigger view. So if we right click, for example, on this, we select the first one and we say move mouse to the other window, then it will actually position the mouse in this main genome window. Another way to accomplish that is just to press the middle button, which allows you to jump between the two views. That is sometimes handy, especially if you are switching on the view or this checkbox over here, where you could slide your mouse across the genome and then it navigates to where you want to be. Well, the first thing we want to actually do with this gene is to show the protein sequence. We can do that by going to show protein sequence, and that then shows the protein sequence with the different amino acids that sit at the exon, at the start of each exon in red. That could also be switched off if you prefer in the settings. And then if you use the square brackets, you could change the width. You could see here the width is changing in the W protein, the width protein box. If you make it wider, each amino acid, then you can see the amino acid number. Every amino acid starts with methionine, which is also the start codon. And if you make it smaller, or making the number of amino acids in each line wider or bigger, and you scroll down, you can see the different amino acids that it's made of. If you hover over any of these, then it highlights the exact position of that amino acid in the zoom DNA view at the top. So you could see if you navigate here to this hydrophobic one, it's going to highlight that hydrophobic one in the zoom DNA view. This allows you to see exactly which DNA is coding for each specific amino acid. So it's interesting. There's this pattern here of amino acids at the end of this protein, which you can clearly see by just hovering over them. If you move your mouse to this one, then you can control the width of that, or you can use your mouse wheel. It's almost like a crossword puzzle. You can switch back to the protein view. Wherever you click, either on the zoom DNA view or on the protein view, when you right click, you get the same menu, which gives you the same options. We've picked this show protein sequence, but you could also say show protein statistics, which will give you the protein statistics for this, which gives you exactly what is the percentage of each amino acid in this protein. and it also gives you like a graph, almost like a fingerprint of what the different percentages of amino acids are that make up this protein. Another very handy view is this when you go to codons, you switch to amino color. You can see that the amino acid colors change in this protein view, which is now not based on its hydrophobic or polar quality, but rather the specific amino acid. If you hover over the amino acids, it shows you which codon was used and what is its polarity. And then if you click on it, there's amino color again. What this is actually the codon usage. Because the last base in every codon can essentially be it can essentially be changed to a different base and then still code for the same amino acid, that which we call a synonymous base. What I've done here is all the codons where the amount of GCs in the codon is more than the amount of A's and T's, I've given it a brighter green color, and the ones where the amounts of A's and T's are more, I've given it a darker color. So that allows you to see the codon bias in a specific organism. It is quite clear that you could get the same proteins in different organisms, but in one organism it uses many more G's and C's as A's and T's, but they still need to code for the same protein. So that the way they accomplish that is by using codon bias. So it's using more G's and C's and still codes for the same amino acids. And you can understand that from the genetic code table. If you look at the genetic code table, here at the bottom you can see that as you go to different columns, the last base changes. So that means that if you have a valine and you move horizontally, it still remains a valine from here to there to there to there. These GTG, GTI, GTT, and GTC, they all code for valine, although the last base in the codon is actually changing. It's the same with proline, it is the same with arginine. And the reason this happens is because the last base in the ribosome actually has a little bit more wiggle room, and that means it is able to make use of a different base. They also call it wobble base pairs. And if you read all of this information that I've provided here, you would be able to make sense of it. So if we just go back to that view again, it shows the same codon bias in the Zoom DNA view. If we switch that back to the hydrophobic view or the properties of the amino acid, and then we right click, there are other things that you can also do. You could also look at the splicing graph, which gives you a view of how the splicing worked in this specific gene. When it is drawn in green, it means it is coded on the positive strand, and when a gene is shown in pink over here, it means it's on the negative strand of the DNA. To simply move between the exons, you simply hover your mouse over the specific exon, and it will go to that exon in the gene. If you zoom out a little bit, you can actually see all the different splice variants. of this gene. And as I said before, you could use the plus and minus to actually pick a specific one. Okay, now that we have this protein displayed over here, you might wonder, how can we actually get a copy of the DNA or copy of the transcript or copy of the protein? So there are a few functions that I've built in, and one of them is when you right click on the protein and you select the specific transcript, you can say here, you can see there's copy the transcribed DNA to the clipboard and plus the comparisons. Okay, so the comparisons window is this window I'm referring to. It's gonna when you close Visual Genome Browser, it actually saves it to disk, so it's like a persistent storage of the clipboard information that you've copied. So let's just go here and we say copy the transcribed DNA to the clipboard. So this is what happens when RNA polymerase reads the DNA and transcribes it into RNA. It says here, copy the transcript before before spliced by spliceosome to clipboard. So if we go and look at how it looks in this comparisons view, you can see here it lists it there, but it has also copied it to the clipboard for you. So if I open a notepad and I paste it, you can see here what it's done is that it has copied the DNA where all of the exons are in capitals and all of the introns are in small caps. So you can see the amount of bases that form part of an intron. So this is what RNA polymerase does. It's just simply starts from the beginning of the gene and it copies all of the bases, including what's in the introns. The introns needs to be removed later on by looking at this this GTAG signals. So this is also available by just selecting the specific entry here and then saying copy. But in this case, it actually makes it formats in a specific form. So you have to copy it directly from the clipboard. If you want to copy, for example, the splice transcript, that is after the spliceosome has removed the introns, then you select this option. And then if you look at how it looks on the clipboard, it's much shorter. This would contain the coding sequence, but there is a 3' untranslated region that still needs to be removed. So if we want to only copy the coding sequence, then you would select this one. And this would always start with ATG, which is the start codon. So if we do that, you can see here it starts with ATG and it runs all the way to TGA, which is the stop codon. If we right click and we navigate to the different exons, let's just zoom in a little bit. I'm gonna just view it like that. If we step through all the exons, there are 30 of them, we eventually get to the last exon. In order to give you an overview picture of this gene and what parts end up in the protein, I'm just going to click on this button on the left, And this will cause the Zoom DNA view to fill the entire right side of the screen. You can see here that the transcription of the gene starts at the beginning of this this green section, and the translation of the amino acids will start at this start codon. And then there's a big intervening sequence or intron, which is also transcribed. All of this is transcribed by RNA polymerase II until the end of the gene. So if we continue scrolling down, you can see we are still going. It's still Pol R2A. This is all transcribed. Now the transcription termination signal comes from this AATAAA signal at the end of the gene. When RNA polymerase II reaches this signal, it terminates the transcription, and then ejects the RNA from the protein. So what happens then is the spliceosome then goes and removes all these intervening sequences, which is shown over here. This is all removed to actually result in the final mature transcript. At the end of the mRNA, a long tail of A's are attached, and that is functioning as a way for the ribosomes to speed up and increase the expression level of the gene. If you want to copy the protein to clipboard, you can then go and say copy protein to clipboard, and then if we paste that here, you can see this is the protein sequence. Okay, I think that is all I'm going to cover today. Thank you for watching my video, and be sure to come back and check out the site to see if there are more videos available.